students, now that you have studied about reproduction in the previous chapter, I'm sure you understand how crucial it is for the survival of living organisms. So, in this chapter, we'll discuss the various aspects of sexual reproduction in flowers in a detailed manner. We usually associate sexual reproduction with animals, but it is just as true for flowering plants, that is, angiosperms, as it is for animals. And in angiosperms, flowers which are morphological and embryological marvels are the sites of sexual reproduction. Students, do you remember the 4 whorls of an angiospermic flower? Let us recall them together. The 4 whorls of a typical angiospermic flower are calyx, which consists of sepals, corolla, which consists of petals, androecium, which consists of stamens, and gynoecium, which consists of the carpels. Out of these, androecium and gynoecium are fertile and are regarded as essential whorls. Thus, we can say that the essential whorls of an angiospermic flower are directly involved in sexual reproduction. In this module, we will discuss the structures and the events in the androecium that occur before fertilization to prepare the flower for fertilization. Now students, we have already mentioned that androecium is the collective term for the stamens. But can you tell me, what are the parts of a typical stamen? Correct. A typical stamen consists of a terminal bilobed anther and a long, slender stalk-like filament. The filament is joined to the anther on one end and either to the thalamus or the petal on the other. Let us now discuss the structure of a typical anther. An anther is typically divided into 2 lobes. Hence, it is said to be bilobed. These 2 lobes are separated by a deep groove in front, but not completely detached from one another. A band of vasculated sterile tissue called connective keeps the 2 lobes connected. Now within each lobe, you'll find a microsporangium at each of its 2 corners. Hence, a typical 4-sided tetragonal anther has 4 microsporangia, and it is called tetrasporangiate. So students, what is microsporangium? Well, microsporangium is a homogeneous mass of meristematic cells that gives rise to the pollen sacs. When a flower attains reproductive maturity, the pollen sacs become filled with pollen grains. These pollen grains arise from the compactly arranged homogeneous mass of meristematic cells that occupy the center of each microsporangium and are known as primary sporogenous cells. For the microsporangia to mature into pollen grains, they require nourishment, which is provided by the innermost layer of the anther wall. This layer, which is also known as tapetum, contains cells that have a dense cytoplasm. The tapetal cells also exhibit polyploidy that they achieve by endomitosis, polyteny, or by forming a restitution nucleus. Now, the anther wall has 4 layers. The layers from the innermost to the outermost are tapetum, middle layer, endothecium, and epidermis. The middle layer is 1 to 3 layered, ephemeral, and degenerates at maturity. The endothecium is a fibrous layer that helps in the dehiscence of the anther due to its hygroscopic nature. The outermost layer, that is the epidermis, is single-layered and protective in function. Now students, do you know how pollen grains are formed? As mentioned earlier, the primary sporogenous cells in the microsporangium give rise to the diploid microspore mother cell. Each of these microspore mother cells forms a tetrad of haploid microspores by meiosis, and this is known as microsporogenesis. During maturation, the anther dehydrates and the microspores dissociate from each other and develop into pollen grains. It is fascinating to know that pollen grains represent the male gametophyte in angiosperms. Now that we know how pollen grains are formed, students, what do you think is the structure of pollen grains? Well, pollen grains are generally spherical structures measuring about 25 to 50 micrometers in diameter. The importance of pollen grains has led to the development of palynology, a sphere of science dedicated to their study. Interesting, right? Pollen grains, like most other cells, have cytoplasm and are surrounded by a plasma membrane. But their primary characteristic feature is their two-layered cell wall, known as sporoderm. The thin inner layer of sporoderm is known as intine. Intine is a continuous layer that is made up of cellulose and pectin. Besides intine, sporoderm has a tough outer layer known as exine. The exine is composed of sporopollenin, which is one of the most resistant organic materials known. As a result, the exine can withstand high temperatures, strong acids, and alkali. It would surprise you to know that no known enzyme can degrade sporopollenin. Exine has also prominent apertures called germ pores where sporopollenin is absent. So students, what do you think is the significance of this exine layer? Well, due to the presence of sporopollenin, pollen grains are well protected from hazardous environments and are even well preserved as fossils. Moreover, the exine exhibits a fascinating array of patterns and designs that are of taxonomic significance. The presence of exine should make pollen grains immortal, right? Surprisingly, the pollen grains retain the ability to germinate on landing on the stigma for a particular period, which is known as pollen viability. This period is highly variable and depends on the prevailing temperature and humidity. The pollen grain that we have discussed until now is still the immature male gametophyte, but pollination occurs at 2-cell stage in 60% of the angiosperms and at the 3-cell stage in the rest. The pollen at the second or the third cell stage is said to be the mature male gametophyte. So, now let us take a look at the vegetative cell and the generative cell formed by the mitotic division of the pollen grain. Students, did you know that pollen grains are of significance to humans as well? Since pollen grains are rich in nutrients, their consumption has been claimed to increase the performance of athletes and racehorses. Moreover, pollen tablets and syrups are also increasingly being used as food supplements. But pollen grains also have their share of demerits. Pollen grains of many species cause severe allergies and bronchial afflictions, leading to chronic respiratory disorders in some people. Some of the plants that cause pollen allergy are Parathenium, Amaranthus, and Chenopodium. Nevertheless, pollen grains play an important role in plant breeding programs for conservation. Hence, they are stored for years in liquid nitrogen at -196 degrees Celsius in a process known as cryopreservation. These storage centers are also known as pollen banks. So students, in this module, we discussed about the structure of androecium, the formation and the structure of pollen grains, and the significance of pollen grains. In our next module, we will discuss about the pistil, megasporangium, and the embryo sac. Hello students, we know that the androecium consists of a whorl of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. Well, in our previous module, we have discussed about the androecium, the male reproductive organs, which are stamens, as a part of the topic pre-fertilization, structures and events. Let us recall the structure of stamen. The stamen comprises of two parts, a long and slender stalk called filament and the terminal, generally a bilobed structure called the anther. Along with this, we also learned about the structure of pollen grains. Now, continuing the topic, pre-fertilization, structures and events, in this module, we will discuss about gynoecium, the female reproductive organ. Well, the gynoecium represents the female reproductive part of the flower, that is, the pistil. The gynoecium may consist of a single pistil known as monocarpellary or may have more than one pistil known as multicarpellary. Generally, the term bicarpellary is used for two pistils and tricarpellary is used for three pistils. When there is more than one, the pistils may be fused together known as syncarpus or they may be free known as apocarpus. We know that the pistil is further divided into three parts. Yes, the pistil is divided into stigma, style and ovary. Now let us briefly discuss the functions of these parts. The stigma serves as a landing platform for pollen grains. It forms the pollen receptive tip, which is often sticky and can be of various shapes. The style is the elongated slender part beneath the stigma, which connects the stigma and the ovary. The basal bulged part of the pistil is the ovary. It is the part of the flower where the seed formation takes place. Inside the ovary is the ovarian cavity called locule. The placenta is also located inside the ovarian cavity. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1, as in wheat, paddy and mango, to many, as in papaya, watermelon or orchids. The female gamete of the plant, known as egg or ovum, is present inside the ovules. Now, let us accustom to the structure of a typical angiosperm ovule. The ovule being a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. Thus, hilum represents the junction between ovule and the funicle. A mature ovule consists of a food tissue covered by one or two future seed coats or protective coverings known as integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Opposite to the micropylar end is the chalaza, representing the basal part of the ovule. Here we use the term nucellus. Now you all might be wondering, what is it? Well, a mass of cells enclosed within the integuments is known as nucellus, and the cells of the nucellus have abundant reserve of food materials. Moving on to the next part of the ovule, we have the embryo sac. The embryo sac is a large oval cell in which fertilization and development occur. The embryo sac or female gametophyte is located in the nucellus. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. Before learning about this process, let us understand the term megaspore. Here, the megaspore, also called macrospores, is a type of spore that is present in heterosporous plants. The megaspore germinates into a female gametophyte which produces egg cells, And the process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell, abbreviated as MMC, in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division, and meiosis results in the production of 4 megaspores. Here, I have a question for you all. Is the megaspore haploid or diploid? Think about it. Well, the answer to it is quite interesting. The megaspore mother cell or the megasporocyte is a diploid cell in plants which undergoes meiosis, resulting in the production of 4 haploid megaspores. And this also explains the importance of the megaspore mother cell undergoing meiosis. That is, the MMC or the megaspore mother cell undergoes meiosis in order to maintain the ploidy of the gamete. In a majority of flowering plants, the megaspore at the chalazal end remains functional while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte, that is, the embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. And as discussed earlier, the embryo sac is a large oval cell in which fertilization and development occur. Now let us study about the formation of the embryo sac in a detailed manner. The functional chalazal megaspore enlarges to form the embryo sac, and its nucleus divides mitotically to form 2 nuclei, which move to the opposite poles, forming the 2-nucleate embryo sac. The nuclei in the 2-nucleate stage divide mitotically to form the 4-nucleate stage. This in turn forms the 8-nucleate stage by dividing mitotically. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not followed immediately by cell wall formation. Hence, we use the term 8-nucleate stage instead of the term 8-cell stage. After the 8-nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or the embryo sac. Observe the image shown here. 6 of the 8 nuclei give rise to 6 different cells when surrounded by cell walls. The remaining 2 nuclei, called the polar nuclei, are situated below the egg apparatus and constitute a single large cell known as the central cell. And there is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes to the synergid. 3 cells are at the chalazal end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8-nucleate, is 7-celled. Now let us move on to the next topic, that is pollination. We have learnt that the male and the female gametes in the flowering plants are produced in the pollen grain and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. So, how is this achieved? Well, here the process pollination comes into action. You might be well aware of this term, right? Yes, the transfer of pollen grains from the anther to the stigma of the pistil is termed as pollination. Also, flowering plants have evolved an amazing array of adaptations to achieve the process of pollination. They make use of external agents to achieve pollination. So students, in our next module, we will discuss about the process of pollination in detail.
Hello students. We know that in flowering plants, both the male and female gametes are non-motile and they have to be brought together for the process of fertilization. And pollination is a process to achieve this objective. So students, in this module, we will discuss about pollination and kinds of pollination, and along with this, we will also discuss about the agents of pollination. Now here comes the obvious question, that what is pollination? Yes, the transfer of pollen grains from the anther to the stigma of the pistil is termed as pollination. Now let us look at the kinds of pollination. Depending on the source of pollen, pollination can be divided into three types: autogamy, geitonogamy, xenogamy. Now let us discuss these one by one. First, let us discuss about autogamy. In this type, pollination is achieved within the same flower. Pollen grains are transferred from the anther to the stigma of the same flower. In a normal flower, which opens and exposes the anthers and the stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity, and also the anthers and the stigma should lie close to each other so that self-pollination can occur. Here, let me tell you something interesting. Some plants such as viola, common pansy, oxalis, and camelina produce two types of flowers that are chesmogamous flowers, which have exposed anthers and stigma, and cleistogamous flowers, which do not open at all. In such flowers, the anthers and stigma lie close to each other. When anthers dehisce in the flower buds, pollen grains come in contact with the stigma to bring about pollination. Thus, cleistogamous flowers are invariably autogamous, as there is no chance of cross-pollen landing on the stigma. Cleistogamous flowers produce a short seed set, even in the absence of pollinators. Now you might be thinking about this term pollinators, right? Well, pollinators are animals which move the pollen grains from the anther to the stigma and helps in the process of fertilization of the ovules. For example, small birds, bees, butterflies, beetles, etc. are pollinators. Here, I have a question for you all. Do you think that cleistogamy is advantageous or disadvantageous to the plant? Well, cleistogamy has a share of both advantages and disadvantages. The advantage of cleistogamy is that the plant can propagate itself under unfavorable conditions. And as discussed earlier, cleistogamous flowers produce a short seed set, even in the absence of pollinators. On the other hand, the disadvantage is that there is self-pollination, which reduces the chance of variation and evolution of genetically superior plants. Now, let us discuss about geitonogamy. In geitonogamy, the pollen grains are transferred from the anther to the stigma of another flower of the same plant. Yes, you guessed it right. Geitonogamy is functionally cross-pollination, involving a pollinating agent. And genetically, it is similar to autogamy, since the pollen grains come from the same plant. And lastly, we have xenogamy. In xenogamy, the pollen grains are transferred from the anther to the stigma of a different plant. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Now, let us move on to the next topic of this module, that is, agents of pollination. Plants use both abiotic and biotic components as agents for the process of pollination. Abiotic components include wind and water, and biotic components include animals. Majority of plants use biotic agents for pollination. Only a small proportion of plants use abiotic agents. Let us begin with pollination by wind. Pollination by wind is more common amongst abiotic pollinations. Here are some requirements which are to be fulfilled for the process of pollination by wind. Number 1. Pollen grains should be light and non-sticky so that they can be transported by wind currents. Number 2. The flowers must possess well-exposed stamens so that the pollens are easily dispersed into wind currents, and large, often feathery stigma to easily trap airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The tassels on it are nothing but the stigma and style, which wave in the wind to trap pollen grains. Along with this, wind pollination is quite common in grasses. Maybe that's why we find grasses in open places. Next up, we have pollination by water. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. Can you recall, where is water a popular medium for fertilization? Yes, you are right. In algae, bryophytes, and pteridophytes, water is a must for fertilization to happen. Anyways, let's come back to this session. Some examples of water-pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses, such as Zostera. Well, it would surprise you to know that not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as in most of the land plants. So, how does this process of pollination by water happen? Well, to understand this, let us take the example of Vallisneria. In Vallisneria, the female flower reaches the surface of water by a long stalk, and the male flowers or pollen grains are released onto the surface of water. They are carried passively by water currents, and some of them eventually reach the female flowers and the stigma and achieve pollination. Now let us take another example in sea grasses. The female flowers remain submerged in water, and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. Did you know that in most of the water-pollinated species, pollen grains are protected from wetting by a mucilaginous covering? Now let us discuss the pollination by animals. Majority of flowering plants use a range of animals as pollinating agents: bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds, and bats are the common pollinating agents. And as discussed earlier, these pollinating agents are termed as, they're termed as pollinators. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Pollination is also brought about by larger animals, such as some primates, lemurs, arboreal tree-dwelling rodents, or even reptiles, gecko lizard and garden lizard. Now here's a fun fact for you. The world's largest known pollinator is the black and white ruffed lemur. They open the flowers of the traveler's palm with their fingers and push their long snouts onto the opened flowers to get that sweet nectar. When they do that, the flower's pollen coats their fur. The lemur then transports the pollen to other flowers and fertilizes them. Isn't that amazing? How smartly everything is connected? Majority of insect-pollinated flowers are large, colorful, fragrant, and rich in nectar. And what could be the reason behind it? Think about it. Yes, animals are attracted to flowers by color and fragrance. The flowers pollinated by flies and beetles secrete foul odor to attract these animals. But don't you think they do this for free? Do you? Nectar and protein-rich pollens are usually floral rewards to sustain these animal visits. When the animal comes in contact with the anthers and the stigma, the body of the animal gets a coating of pollen grains, because pollen grains are generally sticky in animal-pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. But these pollinators have their own tantrums as well. For instance, butterflies prefer flowers with flat petals that act like a landing strip for them to sit on. But given the fact that 1 in every 3 bites of food depends on pollen, now let us have a look at the outbreeding devices. So, what are outbreeding devices? Well, plants have many mechanisms and devices that are employed to promote cross-pollination, and these are called as outbreeding devices. These devices discourage self-pollination and encourage cross-pollination. But why? What is the reason behind it? Well, this is because continued self-pollination results in inbreeding depression. That is, the decrease or loss of fitness and strength, which is mainly caused due to inbreeding. Some outbreeding devices are: number 1, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of the pollen. Number 2, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. Number 3, the third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from the same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Number 4, the fourth device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, meiosis, it prevents autogamy, but not geitonogamy. While in other species, such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, dioicy. This condition prevents both autogamy and geitonogamy. Can you imagine how dull our world would be had there been no variations? Our future flies on the wings of pollinators. So, this calls for some serious attention towards our next topic, which is Pollen-Pistil Interaction. As the name suggests, Pollen-Pistil Interaction tells us about the pistil accepting the right type of pollen. Interesting, right? Pollination does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Often, pollen of the wrong type, either from other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, compatible, or of the wrong type, incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that lead to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. So how does this happen? The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. So, what happens next? Well, after compatible pollination, the pollen grain germinates on the stigma of the pistil and produces a pollen tube through one of its germ pores. The pollen tube grows through the tissues of the stigma and style and reaches the ovary. Now, through this pollen tube, the contents of the pollen grain are transferred to the ovary. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. So students, all these events from pollen deposition on the stigma until the pollen tube enters the ovule are together referred to as Pollen-Pistil Interaction. So, what happens after the pollen tube enters the synergids? Well, now the process of fertilization takes place. But how? Well, after entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, PEN. As this involves the fusion of three haploid nuclei, it is termed triple fusion, since two types of fusions, syngamy and triple fusion, take place in an embryo sac. The phenomenon is termed double fertilization, an exclusive feature of the flowering plants. You might have heard of the artificial hybridization, right? So, what is artificial hybridization? Well, artificial hybridization is the process in which only desired pollen grains are used for pollination and fertilization. Yes, a plant breeder can manipulate the pollen-pistil interaction or even incompatible pollinations to get desired hybrids. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. But how? Any guesses? Well, this is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehisces using a pair of forceps is necessary. And this step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. And this process is called bagging. This ensures that the flower is pollinated by pollen grains obtained from desirable varieties only. When the stigma of that flower attains receptivity, mature pollen grains collected from anthers of the male parent are dusted on the stigma, and the flowers are rebagged, and the fruits are allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen, and the flower rebagged. Now let us sum it up then. In this module, we learnt about pollination, kinds of pollination, agents of pollination, outbreeding devices, pollen-pistil interaction, and double fertilization. By now, we all know that pollinators are often keystone species, meaning that they are critical to an ecosystem. The work of pollinators ensures full harvest of crops and contributes to healthy plants everywhere. For just this to happen, a marvelous bond exists among different natural elements. Humans here as well manage to use their intelligence and create something new and hybrid. Despite the fact that they make possible the life for us, their lives are in peril. Do you know that it is estimated that nearly 40% of them may face extinction in the coming decades? So, it becomes very essential to protect these pollinators in the world of pollution and climate change. By doing this, we will not just save their lives, but our lives as well. It is impossible to imagine our world without pollinators. So students, thank them and meet me in our next module to understand how amazing work they do. Yes, we will learn about the post-fertilization events in the flower. Hey, why don't you find some pollinators in your garden? Make sure the next time you see them, bees, birds, butterflies, please let them in and watch the magic unfold.
Students, do you like flowers? Who doesn't? Flowers are beautiful, and by now you must have gained clarity as to their deeper purpose in life rather than just pleasing our senses. Didn't you? Yes, right. Flowers are the centers of sexual reproduction in flowering plants. And we have already covered that in our previous modules and saw how skillfully each step leads to the next until we have a zygote. But what happens then? Well, after fertilization, a series of events occur in the zygote to develop into a seed and ovary into fruit. So, let's cover that in our current module. We shall examine post-fertilization events in flowering plants. These include development of an endosperm, development of an embryo, development of a seed, and formation of a fruit. Let's make a start with the first topic. Endosperm development precedes embryo development. We have seen how double fertilization takes place in our previous module. Once one of the sperms unite with the egg cell, a diploid zygote is formed inside the ovule, which lies inside the ovary. The other sperm cell merges with the polar nuclei to form the triploid primary endosperm cell. Hence, each ovule contains 2 fertilized cells, and this feature is unique to these flowering plants. So, what happens next? The nucleus of the primary endosperm cell now undergoes repeated mitosis division to form the endosperm tissue with no cell walls. But what is the role of this endosperm? Well, the cells of this tissue are filled with reserved food materials and serve as the embryo's food supply during the early development. So, do you like coconuts? Well, yes, who doesn't? The water in the coconut is nothing but the free nuclear endosperm, which is made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. However, in some plants like peas and beans, the developing embryo consumes the endosperm fully before the seed maturation, and in some cases, it may persist till seed maturation. For example, castor and coconut. So, here is an activity for you. Split open some beans, peas, groundnut, etc. and look for the endosperm. Do you find anything? Now let us move on to the next event, which is embryogeny, or the development of an embryo. This major event takes place inside the ovule of the carpel. But how? How does this happen? The diploid zygote divides many times by the process of mitosis to produce an embryo. The zygote divides to form a proembryo and subsequently to globular heart-shaped and mature embryo. The embryo derives the nourishment from the surrounding endosperm and it continues to grow. It then differentiates to become a plumule, young shoot, radicle, a young root, and either 1. monocots or 2. dicots, cotyledons, which are the seed leaves. It is attached to the wall of the embryo sac by a suspensor. There are some characteristic features of a dicotyledonous and monocotyledonous embryo. In a dicotyledonous embryo, there is one embryonal axis and 2 cotyledons. The portion of the axis above the level of the cotyledons is the epicotyl, which ends at plumule or the stem tip, while the cylindrical portion below the level of the cotyledons is hypocotyl, that ends at the lower end in the radicle or the root tip. The root tip is covered with a root cap. In a monocotyledonous embryo, there is only one cotyledon, which is situated towards the lateral side of the embryo axis. Cotyledon is known as scutellum in the grass family. At the lower end, the embryonal axis has the radicle and root cap, which is enclosed in an undifferentiated sheath called the coleorhiza. Towards the upper side of the embryonal axis, epicotyls is present, which has a shoot apex and a new leaf primordial in a hollow four-layer structure, the cleoptyl. Do you know most zygotes divide only after a certain amount of endosperm is formed? Well, yes. This is an adaptation to provide assured nutrition to the developing embryo. As the embryo and the cotyledons enlarge, they become crowded inside the developing seed and are forced to bend. Ultimately, the embryo and the cotyledons fill the seed, at which point the seed is ready for dispersal. You will be surprised to know that the embryo enters the state of dormancy or inactivity, and it resumes its growth only when the seed germinates. The developing seedling will rely on the food reserves stored in the cotyledons until the first set of leaves begin photosynthesis. Now, let's examine the development of seed and the formation of fruits. Immediately after fertilization, the zygote divides to form the embryo, while the ovule develops a tough coat called the seed. In angiosperms, the seed is the final product of sexual reproduction. A typical seed consists of a seed coat, cotyledon, and an embryo axis. Not only this, but the seeds can be divided into albuminous and non-albuminous, depending on the endosperm consumption during the embryo development. As the name indicates, the former retains the part of the endosperm. For example, wheat, maize, barley, castor, sunflower, etc. While the latter shares its endosperm completely during embryo development. For example, pea, groundnut, etc. Did you know that some seeds can remain in the state of dormancy for as long as 2000 years? Interesting, right? Yes, when the conditions are right, the seed will take up water through the micropyle by the process of imbibition. This triggers the beginning of the growth of the seed. So, can you think of any advantages of the seeds? Well, yes. They not only provide protection to the young embryo, but also being the product of sexual reproduction, they generate various genetic variations. Now, we are very well aware of the fact that seeds and angiosperms are covered. But what covers them is my question to you. Yes, it is the fruit. But how? How are fruits formed in angiosperms? Well, let's see. Right after fertilization, the flower loses its shine. The ovary ripens and the other parts of the flowers, like sepals and petals, wither. The ovary continues to grow until it develops into a fruit. The fruits may be fleshy as in guava, orange, mango, etc. or may be dry as in groundnut, mustard, etc. Now you must be thinking, what is the function of a fruit? Protection of seeds? Am I right? But if we eat them, how are seeds protected? Well, think about it. Fruits aid in seed dispersal. Just like the flowers, they too have a bigger mission to accomplish, which is to land the seeds at proper conditions safely so that a new plant is formed. These fruits are not just whorls for the seeds, but interestingly, they have whorls too. And this is known as the pericarp. So, what exactly is this pericarp? Well, the part of the fruit formed from the ripened ovary is called the pericarp. Although in most of the species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. And such fruits are called? Yes, such fruits are called parthenocarpic fruits. Can you name one such fruit? Yes, you guessed it right. It's banana. What about other fruits? Well, they grow from the ovary only, and so they are called as true fruits, such as guava, orange, etc. Some fruits like apples, cashew, and strawberry develop from the thalamus. Since they do not develop from the ovary, these fruits are called false fruits. And not just fruits, even seeds are formed without fertilization, and this is known as apomixis. Grasses show this phenomenon. Apomix, or a plant that reproduces by apomixis, have several advantages in horticulture and agriculture. Apomictic seeds can be formed in different ways. In some species, reduction division does not occur in a diploid cell, and embryo is formed without fertilization. In some other species, nucellus cells around the embryo sac divide and protrude into the embryo sac and develop into the embryos. So, here's an interesting fact. Humans have identical twins, and just like that, plants too show this alike feature in the name of polyembryony, PE. It can be defined as a simultaneous emergence of two or more seedlings from one germinated seed. Well, that's a lot of information about flowering plants. So now, why don't you just pause the video and recall the journey we've covered so far? Or, let's do it together. Flowering plants or angiosperms are composed of calyx, sepals, corolla, petals, androecium, stamens, and gynoecium, the carpels. Pollen grains or the male gametes are created in the anther of the stamen. A carpel consists of stigma, style, and ovary. Ovary contains the ovule, where the female gametes or the egg cell is formed. Pollinators like bees or abiotic sources like wind and water aid in the process of pollination. Once the pollen grain has landed on the stigma, the pollen tube grows and the union of the sperm and egg cell takes place. Flowering plants exhibit a unique feature of double fertilization. The ovule develops into seed and the endosperm provides the nutrition to the embryo inside the seed. The wall of the ovary ripens to become a fruit and other parts of the flower usually degenerate. And that's how we get a fruit. So students, in our next module, we will discuss yet another interesting topic of biology. So till then, why don't you explore some interesting creations of nature?
We'll begin with a new topic, that is pre-fertilization events. Now in this topic, we'll study what all events take place before fertilization in case of a flower. So very firstly, we'll study about some of the basic parts which are involved in fertilization in a flower. Several hormonal and structural changes result in the development of a flower. So what is responsible for the development of a flower? They are hormonal and structural changes taking place inside it. Inflorescences bear the flower buds and then the flowers. Flowers are the reproductive parts of a plant. So flowers, they are an important part of the plant because they help in reproduction. In the flowers, the androecium, which is the male reproductive part, and the gynoecium, which is the female reproductive part, both of them develop. So flower is the reproductive part of the plant consisting of androecium, the male reproductive part, and the gynoecium, the female reproductive part. Now here in this image you can see the picture of a flower. You can see the ovary being shown, petal, sepal, bract. Then the leaf is shown, anthers are shown, and stamen, it is the male reproductive part which consists of the anther and the filament, as you can see here. And this is the pistil, which is the female reproductive part. So that were the basic points about the various parts of a flower involved in fertilization. Now we'll study about them in detail. We first look over androecium, which is the male reproductive part. The androecium consists of whorls of stamen. So the stamen is what constitutes the androecium. The stamen consists of the filament, which is a long and slender stalk, and anther, which are bilobed structures. Now here in this image you can see very clearly the diagram of a flower showing stamen. You can see this yellow thing, these are the anthers, and the stalk which contains them is the filament. Filament is attached to the thalamus or to the petal. Now as you can see in this image, filament, it is attached to the thalamus or to the petals. Now we'll study the detailed structure of the anther. Now what actually is the anther? The anther is a four-sided, that is a tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. So the anther is a tetragonal structure consisting of 4 microsporangia. As you can see in this image, 4 microsporangia are shown and they are located at the corners, 2 in each lobe. The microsporangia develop further and become pollen sacs. This is very important. The microsporangia, they lead to the development of pollen sacs. The pollen sacs extend longitudinally all through the length of an anther and are packed with pollen grains. Now these pollen sacs, they further extend through the length of an anther and are packed with pollen grains. So this is the basic structure of an anther. An anther consists of 4 microsporangia. The microsporangia further develop into pollen sacs, which consists of the pollen grains. Now after having seen the structure of the anther, we'll study the structure of the microsporangium. In a transverse section, microsporangium appears near circular in outline. So when we cut a transverse section of the microsporangium, it appears circular. It is generally surrounded by 4 wall layers, and they are the epidermis, the endothecium, middle layers, and the tapetum. So what are the 4 layers of the microsporangium? The epidermis, the endothecium, the middle layers, and the tapetum. The outer 3 wall layers perform the function of protection and help in dehiscence of anther to release the pollen. So the outer 3 layers, they help in the protection of the microsporangium, and they also help in the dehiscence of anther to release the pollens. That is, it makes the anther burst open to release the pollen grains. And the innermost wall layer is the tapetum. See these layers in this diagram. Now you can see the outermost layer, the epidermis, being shown. The next layer you can see the endothecium, and the middle layers you can see are shown here, which are present in the middle of this microsporangium. Now these 3 layers, they help in protection and also dehiscence of the anther to release the pollen. Now the innermost layer you can see the tapetum, which consists of these red colored structures you can see. They are the microspore mother cells, about which we'll study later. So this is the basic structure of a microsporangium which is present inside the anther. Okay, now we look at the functions of the innermost layer, that is the tapetum. Very firstly, it nourishes the developing pollen grains. So it provides nourishment to the pollen grains which are developing inside the microsporangium. Cells of the tapetum possess dense cytoplasm and generally have more than 1 nucleus. So the cytoplasm is very dense inside the tapetum, and it generally possesses more than 1 nucleus. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. So when the anther is young, there is a group of compactly arranged cells which are called the sporogenous tissue. They occupy the center of each microsporangium. So this is a characteristic of the anther which consists of sporogenous tissue. So that were the various structures that are involved in the fertilization in case of a flower. Now we begin with the process of microsporogenesis. It's a very important process. It is the process of formation of microspores from PMC, that is pollen mother cells. So basically, the formation of microspores is known as microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells, that is the microspore tetrad. So the microspores, when they are formed, they arrange themselves in the form of a tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. So once the anther matures, the microspores, they dissociate from each other. They had formed a tetrad earlier, now they dissociate from each other and they develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. So what is happening inside the microsporangium? A lot of microspores of the pollen grains, they are formed and they are released once the anther bursts open. That is the dehiscence of the anther. Here in this image, you can see beautifully shown spore tetrads. They are very clearly visible. The tetrads are formed of microspores. So that was all about microsporogenesis, that is the formation of microspores. Now we study about pollen grains. What are pollen grains? The pollen grains, they represent the male gametophytes. So actually the pollen grains are the male gametophytes. They have a prominent 2-layered wall. So the pollen grains have a 2-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. So the outer layer of the pollen grains is very hard and is known as exine, is made up of sporopollenin. Sporopollenin is the most resistant organic material. So exine can withstand high temperatures and strong acids and alkali. So this is why exine can withstand high temperatures as well as acids and alkali, which are very strong. The pollen grain exine, it also has prominent apertures called the germ pores where sporopollenin is absent. So the exine also consists of germ pores and the germ pores do not contain sporopollenin. Now here in this image you can see the enlarged view of a pollen grain which is shown. You can see a hard outer layer called exine being shown with germ pores in it. So that was all about the outer wall of the pollen grain, that is the exine. Now the inner wall of the pollen grain is called the intine, is a thin and continuous layer made up of cellulose and pectin. So it is made up of two macromolecules, that is cellulose and pectin, and it is a thin continuous layer known as intine. Here in this image you can see cellulose being shown. Now in pollen, for some species of plant, the intine is multi-layered. So it is multi-layered in some species of plants and in others it is a single layer. The intine, it lies just below the exine. So exine is the outer hard covering, whereas intine is the inner thin continuous layer. We'll now study the concept of the vegetative and the generative cell. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. So these are the two types of cells which are present in a mature pollen grain. Not a young pollen grain, but a mature pollen grain. The vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. So the vegetative cell, it consists of the food reserve. It is bigger in size and it also has a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. Now here in this image you can see very clearly the vegetative cell and the generative cell. The vegetative cell has abundant food reserve and its nucleus is larger in size as compared to that of a generative cell, which is floating in the cytoplasm of the vegetative cell. So these are the two types of cells present in a mature pollen grain. In over 60% of angiosperms, pollen grains are shed at this two-cell stage. So in case of 60% of the flowering plants, that is the angiosperms, the pollen grains, they are shed at this two-cell stage, that is the vegetative cell and the generative cell stage. Now in the remaining species, the generative cell divides mitotically to give rise to two male gametes before the pollen grains are shed. So in the remaining 40% of the species, the generative cell, it divides by mitosis and it gives rise to two male gametes before the shedding of the pollen grains. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders like asthma, bronchitis, etc. So, this is the harmful effect of the pollen grains. They cause allergies and bronchial afflictions in people, leading to respiratory disorders like asthma and bronchitis.